Okay, this is the second prep for lecture 17. I suspect there will be three videos done. So where I'd left it was talking about when we characterize mutant alleles, we will call them weak alleles if they don't have a very serious phenotype, if they're almost wild type. So they have just a little bit of an effect on the phenotype, whereas a strong allele is one that's really messed up compared to wild type. It's got a very severe phenotype. It's very different from wild type. And when we combine those kinds of alleles with each other, you will find that the weak alleles show dominance over the stronger alleles, which is exactly the opposite of how it sounds like that it should be. So that's why I spent time on it before, and I'm going to talk about it a little bit more here. So this is an example from the fruit fly. The first gene that was ever discovered, mutant allele, was from male fruit flies that had white eyes. And the white gene is found on the X chromosome. And this is a very strong allele, and we also refer to it as a null allele because there's absolutely no pigment produced at all. If you measure, you can sometimes detect a teeny weeny bit, but when you look at the eyes, you just cannot see any color at all. They're just stark white. And then there are a series of different, and this is the strongest allele. Strongest allele. There is an R in here, let's just say. And then right up to, this is wild type, of course, but this one is colored Satsuma, coral. They have all these different names that talk about the amount of pigment. And there's kind of less and less of the red pigment as you work down here. So, white apricot is a sort of a peach color. It's actually very pretty. Cherry is reddish, but it's considerably paler than the wild type, and so on. And it turns out that these function in an allelic series. So you can make flies that are, for example, heterozygous. Females, of course, you can't make males like this, but they have the wine allele and say the white apricot allele. And the white apricot allele is really strong, and the wine allele is very weak because it looks closer to wild type. It's interesting because the percentage of pigment here, these ones they look really low, but you actually can see quite a bit of color in the eye. Your eyes can detect it. And then as we move down the series, it's clearer and clearer that there isn't very much pigment, and you can see it get paler and paler. But when we combine these, we have a protein that's making a fair amount of pigment, and then we have another that's making just a small amount. And so when you combine these, you're going to see the phenotype that looks like the actual weaker allele, and that's what I've been trying to get at when I've been talking about which alleles are dominant over which. And I will have some clicker questions in the class, so we will go through to try to make it nice and clear. There's also a coat color gene in rabbits. I'm probably been thinking about this, and I think that at the end of talking about two genes, I might have a whole lecture that just goes through there's about 5 genes that are helpful to know about for coat color in mammals. All mammals have some aspect of these genes, and they illustrate a lot of the features, things like codominance and epistasis that we're going to talk about, and allelic series and things like that. So I'll probably talk about this C gene again. And so in rabbits, the C, if you have the wild type dominant allele, you have full color, and the color is the pigment is a black color, but there are small yellow flecks on the hair, so they kind of look brown, but really the pigment that they have is black. But there's also the chinchilla allele. The chinchilla allele is recessive to the dominant, of course, and the hair just looks kind of paler overall. And so the furs are white, but they have bits of black in them, and so it has this gray color, and it's called chinchilla. And there's Himalayan. Himalayan is interesting because it's temperature sensitive, and I'll talk about that in a minute, but it's even paler. The fur is pale everywhere except on the extremities, the legs of the body, the nose, the muzzle area, which is a bit cooler, and the ears. So all the cooler spots of the body have darker coloration. And then albino, which is a complete loss of function. So this is the strongest allele, spelled correctly, and this is the weakest of the alleles. But the series works this way. So the wild type is dominant over the chinchilla allele and all the others. Chinchilla allele is dominant over Himalayan and albino. Himalayan is only dominant over the albino, and the albino is recessive to everything. And these show you all the different genotypes that are possible for each of those colors of rabbit. And I should have a picture coming up that shows you. Yeah. So here is a prize-winning rabbit, and you can tell his fur is black. Oops. You can tell his fur is black because when you look at the face and the ears and the legs, you don't have those little yellow flecks in there, but his fur is black. This is a prize-winning, very important rabbit, but he doesn't really care about prizes because you can see he's actually eating his prize here. And then this is what a chinchilla rabbit looks like. You can see that it's sort of grayish all over. The Himalayan, the fur is white over the body, but the places that are cooler, the feet, the ears, and then the muzzle area around the nose and the sort of cheeks, what you might call the cheeks, those are a little bit darker. And then albino. The albino, not only do they not have pigment in the fur, so pure white, but it also affects the eye. So the eyes are pink in albinos, but not in any of the other ones. And I will just point out that there's a lot of mammals have this same temperature-sensitive Himalayan type allele. And it's called different things in different mammals, but this Siamese cat actually has the same kind of mutant allele that this rabbit has. Okay, so strong alleles are more different from wild type, and weak alleles are closer to the wild type, but when we're looking at the dominance relationships, then the weaker alleles are dominant to the stronger alleles. And hopefully this kind of thing is clear. And I will, again, as I said, I will have some questions that we can go over this. And here's just another picture where here's white apricot. It's not making very much of the wild type function. It's got a kind of an orangey red eye. It's probably not even 20%. It's probably closer to 5%. But in any case, you've got an allele where you're making some pigment. The protein that is encoded is allowing for some pigment to be made, and the other one isn't. And so if you've got an allele that's making pigment and you've got an allele that's not making pigment, the result will be pigment, right? So the weaker allele is actually dominant over the stronger one. So I've shown it in a few different ways, which I hope one of these will make sense to you. Okay, so that is an allelic series, and we will talk a little more about some of those alleles when we talk about mammalian coat color. And I will be talking about that Himalayan allele in cats and other animals later on when I talk about conditional mutations. So there are also lethal mutations. Now these are genes that when you make a mutation in them, it has a dominant phenotype, but when you make that dominant allele homozygous, they are lethal. And we'll go through a couple of examples of this. In the lecture, I'm just going to go through this one. So people who work on mice, they will have these pure breeding colonies, and the colonies, they should be homozygous, but every once in a while there's a spontaneous mutation that arises, and this one had yellow fur, and so instead of the dark color you're expecting, the mice had this sort of yellowy, it's sort of a beigey brown, but they called it yellow. Okay, so normal mice basically have black hairs with little yellow flakes on them, little flecks, to give them a sort of a speckly appearance. It helps them to blend in with their background. It's camouflage. But the Y mutation, they don't really have the speckles. They're kind of yellow, kind of all over. And it turned out that this was also recessive for lethality. And so we'll just have a look. So a regular agouti mouse crossed to another agouti mouse, the progeny are all agouti. That is, they're black with the little flecks, and they basically all survive. And so one of the first things that they did with, this is the one actually, when they first discovered the yellow mutation, is they crossed it back to agouti homozygous ones that they got from another colony that they knew were homozygous. And they did this because, of course, they don't know if this is a dominant allele or a recessive allele. And if it's recessive, maybe a lot of individuals in that colony are carrying that recessive allele. So you had to cross to females, probably, from a different colony where we knew that gene had not mutated. And then when they looked at the progeny, they had half that were agouti and half that were yellow. Okay, and in that case, it tells us that this is a dominant allele, because we know that this female was homozygous, and so this male that had this mutation, he must have been able to provide a normal allele to some of the progeny, and then his mutant allele to the others. Okay, and so this was interesting. So, we've got this heterozygous mouse that has this dominant phenotype, the yellow fur, and it's nice to work with things like that because you don't have to do any biochemistry. You can just look at the mouse and know what its genotype is. But when they decided to make a pure breeding strain of the yellow mutant, and they were unable to do it because when they crossed female and male yellow mice to each other, they basically had a 1/3 agouti to 2/3 yellow offspring, and they never were able to. This 2:3 ratio tells you that one category, 1/4 of the progeny died. And we know it's not the homozygous agouti because we used them in this cross, and we know it's not the heterozygotes because they were in these crosses. So it tells us that the homozygous ones must die. So there's a change in this protein that causes the yellow to be expressed over more of the hair than is normal. But when both of your copies are mutants, something else goes wrong, and you have it's not possible for the mice to survive that. Okay, so that's a lethal allele, and then we'll go through another example. You may have heard of Manx cats. There are cats that have a really shortened tail, or there's the T gene in mice. They have a very short tail, and so that acts as a dominant allele for shorter tail for heterozygotes, but you can't make homozygous individuals because in early development of the embryo, the whole posterior end just doesn't develop, and so the embryo is incomplete, and you get this lethal phenotype. Okay, so you can also get temperature sensitivity. So this is an effective environment, and so if we have a case where we have pure breeding red flowers, and we have pure breeding white flowers, and we cross them to each other, and then we look at the progeny. The progeny are all heterozygous, but it turns out that this dominant allele for red flowers, when the light is really intense and the temperature is really cool, that protein works fine. But if it's shady and has warmer temperatures, the protein doesn't work properly. It doesn't make the pigment, and we end up with these white flowers. So we call the cool temperature here the permissive temperature because the protein functions to produce the pigment, permissive temperature, but under warm conditions, this is called the restrictive temperature. And usually we find when you've got temperature sensitive mutations, most of the time they don't function properly at the higher temperature because under higher temperature the protein will start to unfold. It won't maintain its proper structure and it won't be able to do the job. You may know that having a high fever is very dangerous. It can cause brain damage, etc. But what you may not know is the reason for that is when your body temperature goes up really high, the proteins that are needed for proper brain function will unfold. You have a protective system that hangs on to them and tries to protect them for a certain amount of time. But if you have a persistent high fever, you could actually die from it because your proteins that are supposed to be operating the cells of your body start to unfold, and now they can't do their job. They can't bind to the substrate. They can't do the enzymatic reaction, and the cells will start to die. It's interesting. It's quite serious, but it's also interesting. If you think back to when we talked about protein structure and how proteins have to fold the right way in order to do their job, if you heat them up, that disrupts especially the hydrogen bonds and causes them to unfold a little bit. Okay, now I'm going to stop here because I think our next, so this basically is just an explanation of what I've just said, and I will talk about this in the next recording.